great honor and pleasure, and with a bit of trepidation. I haven't seen the final product, but I have such great trust in Mitch Stuckey and Emma Campbell, who have put together a little 6.5 minute, I think it was 6.5 minute video. We're going to use this video as a promotion for the campaign. We'll put it on an online fundraiser website. And tonight is the premiere. So I've seen a rough cut, but I haven't seen the final. So we'll go to that next. Woo!
I had a pretty deep bond with nature my whole life. So as a kid, I spent a lot of time playing outside. If it wasn't raining, or even if it was raining, we're just kicked out of the house for the day. So lots of poking around in little streams and tide pools. So I guess that it was a natural that I went into marine biology when I went to school. I remember one day I saw for the first time an onion cell dividing. It was this coordinated activity where these molecules were causing this dance of these chromosomes where they're condensing and synchronously they're lining up, and they don't pull apart until every last one is lined up. Then they all just separate. So from that moment, I was hooked on molecules, molecular cell biology, which is what became my career. As a scientist, watching the political arena from a distance, I came to terms with the idea that politicians maybe lag a little bit behind, particularly with respect to the environment. Throughout the 90s, that's what I expected to happen with climate change. The data was there, we know the reality. It was frustrating that governments weren't getting their act together, but I kept believing that they would. It was in the early 2000s that it started to hit me that this was getting a bit absurd. I continued to write letters and I continued to show up at rallies, but I became much more active in terms of meeting with my representatives and just more engaged. September 3rd, fall 2014, I was in my office and I had word from friends that Kinder Morgan crews had entered the Burnaby Mountain Conservation Forest and clear-cut 13 trees for their survey. So I walked down to the site and met with people down there and saw what was happening. From that day forward, I would come and sit for a couple of hours in the clear-cut a few times a week. There was a growing but small community of us there, and we would sit on the stumps and think about what was happening to our world. So I wrote about that and had an opinion piece published in a local paper. In response to that, someone had come by and left a 5-pound stack of papers. It was Kinder Morgan, and they were suing 5 of us plus a community organization for encouraging people to get in the way of their ability to work in this conservation forest. It threw my life into chaos. I'm, being chair of the department, running my research lab and teaching, and having to spend all this time in court out of the blue. So after our 3 days in court, the judge actually announced when he would be releasing his decision. So we put the word out. Hundreds of people were on the mountain the day that the decision was announced. The decision was in the favor of Kinder Morgan. We had to get out of the way and let them come in and do what they called their surveying work. There were a number of protesters who spent the night up there. They were prepared that something was going to happen. Indeed, the police came in very early that morning and began arresting people. As soon as I heard it was happening, I came to the mountain and I did witness some of those arrests, and I saw that they were quite aggressive, and people were thrown to the ground, and it was an intense day. So that night, I had a lot of soul searching to do. So I called my son and I said, Jacob, I'm considering putting your entire financial safety net at risk. And more than that, I might not be able to visit you. I might have no financial safety net for you, and I might not be able to visit the United States. What do you think? Should I do this? Should I cross the injunction? Should I get arrested? He didn't hesitate. He said, Mom, yes, you're doing something more important for me than looking out for my financial future or traveling to visit me. This is so much more important and so much more valuable. Thank you for doing this. I support you. So I don't think I got much sleep that night. And the next morning I showed up at the mountain, and the press was there, and I said, you know, I explained what civil disobedience is and the important role that it plays in our society. And I was without any other course of action. I had done every other course of action available to a citizen. All that was left for me was this act of civil disobedience. So I turned and I walked up the mountain and I got arrested. Because of the publicity I got, I received an email from Elizabeth May. And that's where she said, just so you know, I would like you to consider joining me in Parliament. I pretty much knew instantly in my heart that I didn't have a choice, because I can see so clearly the trouble we're in and the enormous injustice that's happening by not taking the serious action that we need to be taking. I am deeply committed. I'm working incredibly hard to be elected because it feels very important to me. There are several really big reasons why the Green Party is the only party that I could have ever run for. Things like participatory democracy, nonviolence, respectful behavior in Parliament. I mean, just everything about the Green Party. And most fundamental, of course, is their respect for and understanding that the environment, the economy, and social justice are all interrelated, and that to create and to work towards a society that I would be proud to live in considers all of those things.
That there was a time decades ago when I was watching onion cells divide and absolutely blown away that these molecules within a cell could organize themselves in some way to make this magic happen, this dance of the chromosomes and this cell division that just to this day still boggles my mind. Although to this day now, at this day, I can stand up before you and we could spend 3 hours and I could tell you a lot about how it works, and it's pretty cool. It turns out that when you get into the molecular machines of a cell, cells in life don't lose their mystery at all. It just gets deeper and deeper and more profound and more amazing. So just my little personal story, I have been studying for a very long time a unicellular green algae. It's a single cell that has a couple of appendages that we call flagella that stick out the top. So it's all one cell with these little guys. You can't see it with your naked eye. You can see these cells under the microscope. And in a warm summer day out in your favorite pond, it might have a green tinge to it. There's a very good chance that that green tinge is my little guys. So these unicellular algae use these appendages, these flagella, to swim through the water. Okay, they're just swimming around in the water column. And they are not quite symmetrical, so they're offset a little bit. So as they swim, they're doing this, they're beating like this, but they're swimming in a helix. So they're beating 50 times a second, and they're doing 1 helical turn, 2 helical turns a second. So they're swimming like this through the water column. Very, why are they even bothering to do this, and how did this come about, and how do they do it? A million questions. Well, the why, they're just like us, just like any life form. The basis, the information is stored, the information that helps the next cell become the same kind of cell is stored in DNA. And just like we know if we're out and getting too much UV exposure in the sun, the cells in our body that are exposed will have DNA damage that can cause mutations, and we can get cancer. Well, if you're a single cell and you get those mutations, that's it, you're done, right? So you no longer divide, you no longer have progeny, it's over. So the cells that have survived have learned to avoid that super bright light. But at the same time, this is a green algae cell. So that means that it has a big central chloroplast. It makes its living by converting sunlight to energy. It's not eating anything else, it's eating the solar energy. So I kind of love the, anyway, we'll get to the solar energy in a minute. It's taking that solar energy. So it has to have enough light for that. So it has to find this sweet spot. Not so much that it's going to get UV damage, not so little that it's going to starve. So it has, on one side of the cell, has evolved this amazing structure that we call the eye spot. And it has these carotenoid pigments, these orange pigments in these fat molecules that form a little cup. And what that does is it shades, this part of the cell membrane is shaded from this side. So this tiny little cell only gets light from one side at that patch. A good friend of mine, one of the wonderful things about science is you develop wonderful friendships all around the globe. So I've been working in this field for 30 years, and a friend that has also been doing that for 30 years, and we see each other every year at conferences. He discovered the molecule that is in that region of the cell that senses the light, and it turns out it's very closely related to the molecules in our eyes that sense light. So evolutionarily speaking, humans are over here, my green algae is over here, and you have to go way back in time to the first cells that had a nucleus before you find our common ancestor. In spite of that, we have these molecules that are almost identical. So these molecular machines that work, nature keeps those, right? We just add new things to those. So here we are. We can detect light on this side. We're swimming in a helix. Voila, radar. The light is coming there. It's really bright. It's hitting. I get a beep. I'm swimming. I get a beep. And how loud that beep is tells me whether that light is too bright or not bright enough. And I know I can adjust, and I don't beat quite so hard with this one. I beat harder with this one, and I need some more light, so I'm swimming this way. So this guy can maneuver. So for the past 25 years, I have been studying these things, these flagella. We know that every time a cell divides, it grows new ones. And it swims and lives for a day, and then it's time to divide into two cells because it's been harvesting lots of light, it's been growing, it's getting fat, it's time to make two where there were one. But to do that, it has to pull these machines back in, and I wish we had an hour so I could tell you why and how that works, because it's really cool. So it does that, and that's what I've been studying. And I've been studying it because I just think it's really cool and interesting, and the machines are cool and interesting. And one of the discoveries we made was that one of the genes we discovered that was important for that process was also mutated in a mouse that people had been using to study polycystic kidney disease. This is a strain of mice that developed polycystic kidneys. No one knew why, but they were using this strain of mouse to test various drugs because this is a disease that is very common in humans. We don't really understand it. We understand it now a lot better than we did, but we still don't have great treatments. But this is the wonderful thing about science. A different friend, who had also known for a long time, had discovered another gene at the same time. Actually, he was ahead of me by a year, and his gene caused these things in the chlamydomonas. It caused them to grow only halfway. So they got short ones, and they couldn't swim very well. And when he looked, he found a mouse that had polycystic kidney disease with the mutation in the same gene. And then we found another one, and we had another mouse. There were only 7 mice in the world. There were only 7 strains that had polycystic kidney disease, and all of them seemed to be related to these structures, which no one really quite had paid attention, but we went in and looked. Greg went in and looked, and Greg found that our kidneys and mouse kidneys have very short versions of these that we thought didn't do anything. It turns out they're critically important for processing information about the environment of those kidney cells. So they were sending the wrong signal to the kidney cells, and the kidney cells were forming cysts instead of tubules. So we know a whole lot more about polycystic kidney disease. Other discoveries that we've made more recently have led us into the field of cancer. So we're starting to understand how this cycle of shrinking and extending these flagella is related to cell division, and it turns out that that relationship is probably also happening in human cells. There is no way, when I started doing this work, no way anyone could have predicted what the outcome of our research would be. We have recently finished another genetic screen where we've identified 4 new genes. We don't know the implications of that yet. We don't know what that might lead us to. But right now, Andrew was generously talking about the funding in my lab. It used to be true.
Mine too.
Used to be true. My lab, up until recently, and well, until right now, although I'm letting it go, my lab is now funded at 25% of what it was funded at when Harper came into power. Funding for basic research has evaporated in this country. If I want to get the kind of money that it takes to do this research properly, I have to find an industry partner. Now I ask you, can you name an industry partner that might be interested in somebody who's studying unicellular algae and how they swim through the water? Right? This is curiosity-driven research. This is discovery science. This is the stuff that feeds the pipeline of innovation. Our current government is looking way out here at what's right there ready for innovation, and they're calling it science, but it's very, very applied. We always need a balance, so I'm not criticizing that some money is out there, but it's absolutely inappropriate to put it all out here or huge amounts of it out here because the pipeline will run dry. And we're not going to notice it for a while, but I'm closing my lab. I made the decision my grant is due for renewal in November, and I've made the decision I'm not going to apply. At the level of funding that I have right now, we are below a critical mass in the lab. Science works best through collaboration. It works best when there are people in the lab that bring different expertise. They talk to one another, you brainstorm, exciting new things happen. I've been struggling for years. It's not fun anymore, and it's not efficient anymore, and we're not competitive anymore. We're not able to keep it going. So I managed, I'm getting through this without breaking down. That was always the risk when I talk about my science. It's very hard to go. It's been my passion for a long time. So it's not just that they're doing basic research. They don't want to hear about science. They don't want to know about the real world. There is, in Stephen Harper, and unfortunately in many other people who have entered politics and have found themselves in positions of power, there is a feeling that they know the best. They already know the answers. They have an ideology that they are convinced that they know what is right for human society, they know what is right for the world, and they're not open to hearing otherwise. They don't distinguish opinions and points of view from reality. And as a scientist, one thing you learn really early is that the real world always trumps your ideas. Most of your ideas really are garbage, right? And as a scientist, you get that feedback all the time. So, you know, your experiments are telling you, no, you might have thought that was a cool idea, but it's wrong. And in science, you know that. You get that feedback. In politics, if they would listen and pay attention, they would be getting that feedback as well. But they're so ideologically driven right now that they're not hearing that difference. So I was motivated to make this step because there isn't room for my science in this country anymore. That is a concern not just because we're losing the pipeline of innovation and of new ideas and new discoveries, but we're also not training those students. The students that would have been in my lab are now going into other fields. They're not going into science. The science is becoming concentrated, what's left of it, in the big universities, in UBC and University of Toronto, and places like UNBC, where they have been training scientists, young people who may not move to the big cities right away. Those places are losing their funding faster than anyone because it's harder to stay competitive when you're not part of an ecosystem where the kind of research is happening. So I'm going into politics for science. I'm going into politics to get an evidence-based democracy back. That's what got me going. So as you heard in the film, I was really waiting and waiting and waiting for governments to kind of step up and take action on climate change. The data is obvious. But it's not happening, it's not happening. And at this point, we are already, I'm stunned at the state. I don't want to bring everybody down. But the reality is, our oceans here, the Salish Sea, is already seriously acidified. So cold water absorbs more CO2. And you can think of that when you warm a pot of water on the stove, the bubbles form before it boils because warm water doesn't hold as much gas. Cold water holds more gas. Cold water here is holding more CO2. That CO2 is becoming carbonic acid in the ocean, is becoming acidic. And organisms, the plankton that form these calcium-based shells for the larvae of shellfish, or just for those species that live in the plankton, we're losing species at shockingly fast rates. So both shellfish species, planktonic species, and as the species composition of the plankton changes, that will go all the way up the food chain. So Eddie was talking about changes to the salmon. These changes will also be affecting the salmon. The salmon are already stressed. Changes in temperature of the rivers, changes in acidity of the ocean. These are all adding to the stresses of these fish. Okay, not to be too much of a downer. So, getting from science to climate change, being a climate change activist, led me very quickly to understanding climate justice. Intergenerational justice, global justice, and the more I looked into it, the more that I began to understand how out of control the globe is at the present time. The more I learn about things like free trade agreements and the fact that multinational corporations have seriously, I used to think this was a radical, out of touch with reality kind of thing to say, but frankly I now know it to be true, that we have seen a corporate coup d'etat. Most countries have been taken over by corporations who have taken our sovereignty through these free trade agreements. So examples would be a recent example, and I'm going to just pick on the NDP for fun, just one party. They have now come out, and Thomas Mulcair is bullish on free trade agreements. Most recently, the NDP voted 100% in favor of the Korea-Canada trade agreement. That trade agreement means that Korean companies that have rights on mines, coal mines in Vancouver Island, if we try to put in environmental regulations to protect our waters, to protect the oceans and the streams and the environment, we will be sued by Korea for interfering with their abilities to make profits. And that's just a little tiny taste. Many of us were at a free trade session last night where we heard a couple of hours of examples along those lines. So we have to step up. The first thing with respect to things like the free trade and what is happening in the world is that people need to recognize what's happening. The TPP, why is it being kept secret? It's being kept secret because it's full of stuff that if people knew about it, they would be shouting in the streets that this cannot happen. So we need to do that. We need new government. We need people that are not, as Eddie was saying, and as Andrew was saying, who are not just looking for the next election. People who actually deeply, passionately care about our future. And so the Green Party at this stage, we're doing great, and Andrew, thank you. It's a green revolution happening, and it's very exciting to be part of it. But however great we might be doing, we're not so disillusioned, or not so out of touch with reality to think that we're going to form a majority government.
Federally.
Federally. Yeah, okay, just federally. However, I don't want anyone to have any doubts about the power that a Green MP can have. The Green Party will stand up for you. It is in the Green Party policy that Green MPs' first allegiance is to their constituency. I will be your representative. I will hear you. I will take that perspective to Parliament, and that is the way Parliament should work. If we had a house full of MPs who brought to the table their perspectives and were willing to listen to evidence and human perspectives, values. Across this country, we have differing values, but the data is the same. So we take that data, we listen to those values, we talk about them, and we say, this is what's best for the country without screwing this region of the country, right? So we look at everything, and that's how it should work. Green MPs will help make that happen. And I firmly believe, and I learned this on Burnaby Mountain, that when you have the courage and you stand up and you say, yeah, they might sue me, they might take my house, they might take my retirement savings, so what? That stuff doesn't matter compared to these other issues. And when you do that, you'd be amazed the people that stand up with you and support you and say, yes, we can do this. And that's what Green MPs will do in the House. We will say, hey, you guys with all those different colors, you don't have to be whipped. Stand up. Speak up for your constituents. Be a representative. And so we can start to take back the House of Commons for the people.